Hello everyone and welcome back for more biotechnology. So in the last chapter we basically got a good handle on what DNA is in terms of structure and function, and then different ways of studying DNA with a particular focus on gel electrophoresis. So in this chapter we're going to do the same sort of thing, except this time we're going to be looking at protein in very much the same way. We're going to get a survey of protein structure and function, how are proteins produced and maintained in the cell, and then, just as we did with DNA, we will talk about how we can study proteins by using very similar gel electrophoresis techniques. So let's go ahead and get into this. So if you'll recall from Chapter 2, in which we talked about how all different types of biomolecules are put together, you'll remember that proteins are polymers of amino acids in the same way that DNA and RNA are polymers of nucleotides. One of the big differences, though, is that whereas DNA and RNA are made up of 4 different types of nucleotides each, proteins are going to be a little bit more complicated because there are up to 20 different amino acids that we can use to build proteins. So if we look at an amino acid structure, every amino acid is going to look the same with the exception of its R group. So every amino acid is going to have an amino group, a carboxyl group, and then finally that variable R group. So each one of the 20 amino acids looks exactly the same, but each one has its own unique R group, which is going to give that particular amino acid its own chemical properties. So depending on what the nature of this R group is, it can cause the amino acid to either take on a hydrophobic nature, in which the side chain shies away from water. The side chain can be hydrophilic, in which it likes water. The side chain can be either acidic or basic. If it's acidic, it's going to take on a negative charge at a neutral pH. If it's basic, it'll take on a positive charge at a neutral pH. Or these R groups can be what is called aromatic, which isn't going to mean a whole lot to us just yet. But basically, aromaticity is referring to these ring structures in which the electrons move around in a resonance. So like I said, that doesn't mean a whole lot to us, so don't worry about it too much. So the idea here is that when the protein eventually folds into its final 3-dimensional structure, which we're going to talk about a little bit later, it's going to do so in such a way that it should satisfy as many of those side chains as humanly possible. So we already kind of have an idea of, for each side chain, depending on what that side chain is, what would make that side chain happy. So for example, any polar side chains are going to want to participate in hydrogen bonds, usually with water. So these are those hydrophilic side chains. So they want to be with water, so when the protein folds, we're going to get them as close to water as we can. Any acidic or basic side chains will want to interact with each other. The negatives and the positives will want to attract each other, just like in the way static electricity works. Those hydrophobic side chains that don't like water, well, we're going to try to satisfy them by burying them in the core of the protein so that they don't have to see or interact with the water that surrounds the protein. And then the aromatic side chains, without getting into too much detail, they like to kind of stack on top of each other like Pringles. So for some reason, that just seems to make them happy. So regardless of what type of side chains are present in a protein, the protein is going to eventually want to fold in such a way that makes all of these side chains happy. So you can see in this picture a model of what a protein might look like after it is folded into its final structure. So you can actually see some of these side chains here modeled in purple, and you can see some of these side chains are pointed at each other, some of the side chains are kind of pointed away from each other. So whatever the 3-dimensional shape of the protein ends up being, it's going to be done in such a way that all those side chains are as satisfied as possible. So the way you actually end up with a structure like this is by using a technique called X-ray crystallography, which is not something that we're really going to talk about in this class. But essentially the idea of how something like this works is that Once you have purified a protein, you treat it in such a way to try to get the protein to fall out of solution and form as pure of protein crystals as possible. And then essentially you take those crystals and you basically just bombard them with X-rays. And based on the structure of the protein will diffract the X-rays, which will be picked up by a sensor. And then this allows us to essentially kind of work backwards and figure out what the three-dimensional shape of the protein is based on the diffraction pattern of the X-ray. So, not something we're going to get into in any great detail, but it is definitely something that is used in order to figure out what a protein looks like three-dimensionally. So as we said, when you're building a protein, you are essentially making a big polymer of individual amino acids. So we're basically sticking amino acids together like we're building beads on a string. So when a protein is synthesized, we join together amino acids. We stick them together by forming a type of bond called a peptide bond between the two amino acids. This bond is formed between the carboxyl end of one amino acid and the amino end of the other amino acid. So in this picture that you're looking at here, you can actually see three different amino acids here. So one here in the middle, and then one here on the end. So each peptide bond is formed between the carboxyl end of one amino acid and the amino end of another. Carboxyl end of one and the amino end of another. So for these three amino acids, there are two peptide bonds joining them together to form what we would call a tripeptide, a small little chain of amino acids that contains three of them. So when we're building a protein, we just do this again and again and again, and we make this chain of amino acids as long as it needs to be. But regardless of how many amino acids are incorporated, the final protein product is going to have a free amino terminus or N-terminus, so you can see it over here, and it's going to have a free carboxyl terminus or C-terminus over here. So regardless of how many amino acids we add on to the chain here, we are still going to have a free N-terminus and a free C-terminus because those are actually not actively participating in peptide bonds, so they are free to kind of hang out. Meanwhile, the R groups of each incorporated amino acid will just kind of hang off to the side. So in this particular case, you can see an R group here, you can see an R group here, and then you can see an R group here. They're just kind of hanging off to the side, meaning that what isn't an R group, what actually makes up the backbone of the protein, can actually kind of twist and turn and fold around to try to get those R groups into a configuration that they are satisfied with. And that's going to be what the whole protein folding issue is all about. So when you're looking at a protein in terms of its linear sequence from the N-terminus to the C-terminus, this is called the protein's primary structure. It's basically just a readout of which amino acids are incorporated into the protein and in which order. So that's what's called the primary structure of the protein, the same way that we could look at a DNA strand in terms of its nucleotide sequence. So something that you will recall from earlier in this semester back in Chapter 2, when we're trying to decide which amino acids to incorporate into a protein and at which place in the primary structure, this is all determined by two different things, the genetic code and then also the sequence of nucleotides in both the DNA and the mRNA transcript following the transcription part of the central dogma. So this genetic code table that you're looking at here tells us which codon or which stretch of three nucleotides in a messenger RNA molecule will instruct which amino acid to be incorporated into the protein. So if we look at this mRNA transcript here, read from 5' to 3', AUG, CAG, GCU, and so on, I have them split up into groupings of three nucleotides because that's what a codon is. It's a grouping of three nucleotides. We can actually interpret this and we can match it up with what we find in the codon table of the genetic code, and we can determine from the messenger RNA instructions which amino acids are going to be incorporated into a protein. So as we go from 5' to 3', that will tell us which amino acids make it into the protein from N-terminus to C-terminus. So AUG is going to conform to methionine, CAG is going to give us glutamine, and so on and so forth. So the protein here goes from N-terminus to C-terminus, methionine for AUG, glutamine for CAG, alanine for GCU, phenylalanine for UUU, isoleucine for AUA, serine for UCC, arginine for CGG, lysine for AAA, tryptophan for UGG, and then the final codon, UGA, you will notice is one of our stop codons. So that is the signal that tells the ribosome, as we will find out later, that is the signal that tells us to stop making the protein. Basically, it's a signal that tells you you are done. So this would be the final protein from N-terminus to C-terminus. So the big takeaway here, if you take away nothing else, is that the sequence of amino acids in a protein, meaning the primary sequence of the protein, it is determined 100% by the sequence of nucleotides in the DNA. So this is why we say the gene in the DNA and in the messenger RNA, it's the gene that provides the instructions on how to make the protein. So basically you can think of the DNA as a set of blueprints that it tells you exactly how to build your protein, which amino acids to use, and which order to put them in. Okay, so the primary structure of a protein essentially is just a linear straight chain of amino acids, but the problem with that is that linear proteins don't have any function. So if you take a protein and you just stretch it out in a big straight chain from N-terminus to C-terminus, that protein's not going to do anything for you. So in order to function, proteins are going to have to fold into a mature three-dimensional structure. So proteins have to have a three-dimensional shape in order to do what they are supposed to do. As we've already seen, how the protein eventually folds into its final three-dimensional shape, well, that's all going to depend on the sequence of amino acids and which side chains like to interact with which other side chains. So the first thing for us to talk about going beyond the primary structure is what is called secondary structure. So secondary structure represents the first step in the folding process. So in this step, hydrogen bonds will actually form between the oxygen and nitrogen atoms of the backbone and any stray hydrogen atoms that are sticking off from the amino groups. So the polar nature of the oxygen and the nitrogen atoms will attract the hydrogen atoms, and this is going to cause the backbone of the protein to bend and fold into particular shapes. So this one that you see here is called the alpha helix. So you can actually see that we are twisting the backbone of the protein around and around and around, kind of like a spiral staircase, and it is the hydrogen bonds represented as the dotted lines that is holding the backbone into this particular configuration. So in this three-dimensional space-filling model that you see here in the bottom right, you can actually see this being done in a number of different places. You can kind of see that twisting and turning there, and that represents the alpha helices that are found within that protein. An alternative secondary structure that can be formed, which is also facilitated by hydrogen bonding, is called the beta sheet. This is the same sort of thing, except instead of twisting around in a helical structure, you actually see the backbone of the protein twisting around and around and around in kind of an anti-parallel fashion, and the hydrogen bonding between the two different strands of protein actually hold this structure together here. So what we're going to find out is that different stretches of amino acids within a whole protein are going to want to tend to form different secondary structures. So the idea there is that within a big protein, you may find certain regions that like to form alpha helices and certain regions that like to form beta sheets. And you'll find that all within the same protein. It's just going to depend on which amino acids you happen to find there. So the final step in the protein folding process is called taking on a tertiary structure. So this goes even further beyond the secondary structure. So tertiary folding, as we discussed before, is facilitated by the interactions between the R groups of the amino acids that make up the protein, whereas secondary structures is facilitated by interactions between the atoms that actually make up the backbone of the protein. So these are the rules that we laid out before, so it bears mentioning again. So in order to go from secondary to tertiary structure, you're going to get these side chains to interact in the way that they are satisfied that we laid out before. You want the hydrophilic polar side chains interacting with water. You want the acid and the base side chains interacting together and attracting themselves electrically. You want the hydrophobic side chains buried away at the center of the protein and away from the water surrounding, and you want those aromatic side chains stacked on top of each other. One other way of getting tertiary folding to occur that we didn't talk about before is by forming what are called disulfide bonds. The reason why we didn't mention this before is because there's only one type of amino acid side chain that can participate in this type of bond, and those are called cysteine amino acids. The side chain of cysteine contains sulfur atoms, and when two cysteine side chains interact together, they like to form this type of bond in between them, which is called the disulfide bond. So if you have two cysteine side chains at different points in the primary structure, even if they're fairly far away, the protein can bend and fold in such a way to get these side chains together, and when this disulfide bond is formed, it can cause the tertiary structure to stabilize, and the protein will become a lot more rigid, meaning that if the protein were to unfold, it would be because you had to break that disulfide bond, which can only be done with some difficulty. So disulfide bonds are also important in the formation of what is called quaternary structures. Now, quaternary structure does not actually refer to the way one single protein folds. I already mentioned that the final folded structure of the protein is its tertiary structure. So a quaternary structure actually refers to multiple different proteins coming together to form a large multi-protein conglomeration. A good example of this is hemoglobin, which you can see here in the bottom right. Hemoglobin is actually made up of 4 different proteins called 2 alpha globin proteins and 2 beta globin proteins. Each one of those 4 proteins went through the same folding sequence. It went from primary to secondary to tertiary structure. So for each one of those 4 globin proteins, the tertiary structure represented its own individual final folded state. But when these 4 proteins come together to form the native hemoglobin structure, that is when we start talking about the whole quaternary structure. A good example of this is what we see in types of protein complexes called antibodies. Antibodies, or immunoglobulins as they are also known, they are quaternary proteins that consist of a total of 4 different individual polypeptide proteins, 2 light chain polypeptides and 2 heavy chain polypeptides. We can see here that these heavy and light chains are held together by disulfide bonds, so you can actually see the sulfur atoms that are joined together here. So we have, in this particular immunoglobulin, we have a total of 6 different disulfide bonds that are holding the whole thing together. So we're not going to talk a whole lot about antibodies just yet. We will talk a little bit about them, but are going to find out in the next chapter that antibodies are very, very critically important in biotechnology applications that involve protein detection. So antibodies, if we look at the anatomy of one, antibodies have a variable region up top there that differs from antibody to antibody. So in your bloodstream, you have millions of different antibodies, and they're all going to be a little bit different in terms of what their variable region looks like. The reason for this is that the variable region contains what is called the antigen binding sites that determine what the antibody is going to look for and bind to. So these antigen binding sites are specific for what is called an epitope. An epitope is a molecule or a portion of a molecule to which a specific antibody binds. So if an antibody, say, is specific for and binds to the first 15 amino acids of a protein, then that stretch of 15 amino acids represents the epitope that that antibody wants to bind to. On the other hand, antibodies also have what is called a constant region. The constant region does not actually bind to any antigens or any epitopes. These constant regions are going to be the same for every antibody that comes from that particular species of organisms and for that particular antibody type. The type of antibody you're looking at here is called IgG, immunoglobulin G. So for every human IgG, regardless of which antibody it is and what the variable region looks like, that constant region is always going to be the same. And this is going to become important in protein detection once we get there in Chapter 6. So if we take just a quick little look at an antibody application, let's look at one called ELISA, which stands for Enzyme-Linked Immunosorbent Assay. So this is going to utilize the power of antibodies to detect very specific molecules or epitopes. So the way this works is that you have a 96 well plate. So it's just this little plastic dish that has 96 little plastic wells in it. So the way this works is that you are going to take a mixture of different antigens and you are going to seed them to the bottom of the well. So essentially whatever you have, whether it is materials from a cell or some sort of diagnostic application or someone's blood or someone's serum, you're going to put it in the wells of this 96 well plate, with the idea being that somewhere within that material that you put in the bottom of the well, there is some molecule that you suspect is there, something that you want to detect if it is there. So after these molecules have their chance to stick to the bottom of the well, you are going to then overlay antibodies against a particular antigen or epitope. So if the serum or whatever it is you added to the well initially, if that actually contains the epitope or antigen that that antibody binds to, those antibodies are going to stick to it and not anything else in the well. And then finally, after this is done, another antibody is added that is specific for the constant region of the initial antibody that you added. The initial antibody is called the primary antibody. The primary antibody recognizes the antigen present in the well. The next antibody is called the secondary antibody, and it recognizes the primary antibody. Also noteworthy, the secondary antibody is linked to an enzyme that, when you add a particular chemical, will produce a certain color. So the way this assay works is that once you have done all the steps, if the antigen is present in one of the wells, then you should see a color change within that particular well, and that will give you an idea, diagnostically speaking, of whether a particular antigen is present or not. And depending on how stark the color change is, it should give you a quantitative look into how much of that antigen is actually present. Okay, so that is going to do it for this particular video. So thank you for your attention, and I hope you will join us next time, and we will start talking about the cell biology behind how proteins are actually made in the cell. We have looked at it in a little bit of detail before, but we are actually going to get a much better idea of it in the next video. So thanks again, and I will see you next time. Bye-bye.